Today we are talking about how the virus gets into the cell and then how it can replicate once it does that. So here we see up here at the top we've got our virus. So it's wrapped in that capsid. We talked about the capsid yesterday. The capsid are these proteins that basically are encapsulating or wrapping around, protecting the genetic material, which could be DNA or RNA, single-stranded or double-stranded, as we mentioned previously. So this one in this diagram looks like it's RNA. This one is wrapped in an envelope, and sometimes the envelope is actually helpful to be able to get it inside of the cell. And so once it gets inside of the cell, that's broken down. There's also going to be enzymes that will break these proteins down inside the cell. And when they do, because obviously this cell has not yet recognized that it's bad, and so they'll break down those proteins. And when they do, it releases the viral genome, the viral genetic material, which in this case, again, is RNA. So we've got that RNA there in the cytoplasm of the virus. And just like when we talked about in our last unit with gene expression, once that RNA is there, it is going to be expressed. And so essentially that's what happens. That RNA is converted or used to make mRNA, which is then copied to be able to make more copies of the RNA that will be put into new cells. And then it's also expressed to make the proteins. And then the glycoproteins are also expressed as well. Now, they're a little bit more complicated proteins, so they're made in the ER, just like we talked about with gene expression. Ones that are going to be going to the surface of the cell, they are made in the ER. And so that's what happens here. It makes them to the ER, it sends them in a transport vesicle and takes them to the surface of the cell. And then as these new viruses are released, this is exocytosis here, the way they kind of just bud off and are released, these glycoproteins will surround them and cause that release of that new complete virus, completely made by the machinery and the organelles of the host cell. So lots of viruses replicate this way. The common cold replicates this way. I think flu replicates this way. Many viruses replicate this way. So viruses, when we look at them, are broken down into 6 classes, class 1 through 6. So we're going to talk about one specific class. You don't have to know all the classes, but I do want you to be familiar with this class, though you don't necessarily need to know that it's 6. But they're divided into these classes based on whether they have DNA or RNA, whether it's single-stranded or double-stranded, and then basically how they replicate. And so retroviruses are the specific class that we're going to talk about today. So when you think about that term retro, I'm going to try to break this down for you, so hopefully it will help you remember it later. If you think about retro, when I think about retro, I think about like the 80s, like way back, like going backwards. And so that's what we're doing here with this retrovirus. It's going to kind of work backwards. Okay, so it has the most complicated replication, and it has an enzyme called reverse transcriptase. Again, we know it's an enzyme because it ends in ase. And so this term here, transcriptase, should look like another term that you're very familiar with, which would be transcription. So if you think about what transcription is the conversion of DNA to RNA, right? Specifically mRNA, okay? But we're just going to say RNA in general. So reverse transcription would be the opposite of that, converting RNA to DNA, okay? So in retroviruses, their genetic material is RNA. And so what they do is that genetic material, that RNA, is used with reverse transcriptase to be converted back into DNA. And then that DNA becomes integrated into the host cell. Here I have animal, but into the host cell. And when it becomes integrated into that host cell, it forms what's called a provirus. Okay, so let's look at this image here. So we have one of these class 6 viruses, the virus that cause HIV and AIDS is an example of that. So in it, it's got that capsid that surrounds the genetic material, and the genetic material is RNA, okay? We also have this little green molecule, which is the reverse transcriptase. So it gets inside of the cell. The capsid's broken down, and so the RNA with the reverse transcriptase is released. And so that RNA is going to be converted into DNA. A DNA-RNA hybrid at first, and then that's going to replicate and make the full-on DNA. And so that DNA, this is a host cell, and remember previously I said it was an animal cell, so this doesn't happen in plants. It does happen in like eukaryotic animal cells, which we are animals too. Obviously, HIV infects us, but we are considered in, kingdom Animalia. And so that viral DNA is embedded into our chromosomal DNA. And I'm using our as humans that have HIV, okay? And so then we have, that's what's called our provirus. When that viral DNA gets embedded into the normal chromosomal DNA, that makes up our provirus. So then that provirus, every time the DNA copies itself and the cells are replicated, that provirus grows, okay? And you have multiple copies of AIDS. It starts with just one cell and it replicates. And it also makes, it expresses genes from that virus. And so you see the new viruses being formed here. The mRNA is used to make the proteins and the glycoproteins again in the rough ER, which are then essentially used to make the new virus that's released there. All right, so that's an example of retroviral replication. So it's just a little bit different because it works backwards and works from RNA to DNA and then back to mRNA. Okay. Oops. So we have, obviously, in modern science, we have developed vaccines for a lot of disease. And so vaccines are, just a derivative of the disease that causes the immune system to spike a defense against that disease. So, for example, with the COVID vaccine, since that's what's, right here, right now, the coronavirus vaccine is an mRNA vaccine, and it is the first one that's ever been released of its kind. Now, they've been working on the science behind an mRNA vaccine for a while before COVID, but they never actually made and marketed an mRNA vaccine. And so if you look at the COVID vaccine, what it is, it's an mRNA. So here's my mRNA that I'm going to try to draw a little something of this. And it is encased in a lipid. Okay, remember the lipids. You've seen those before. Hopefully you remember where you've seen those. And I should draw these a little probably more tightly packed together, but for the simplicity of my drawing, I'm just drawing them spaced out. So it's encased in a lipid membrane. Okay, now think about why that lipid might be important for enabling it to get into the cell. Okay, the cell membrane, remember, is also made up of a phospholipid bilayer. And so in chemistry, and I don't think you all did this last year, for those of you all that had me for biochem, we didn't get quite that far. But what I'm just teaching my students in here right now is like dissolves like. Things that are lipids can move easily through lipids. We talked about that kind of when we talked about transport. We talked about how polar things that are hydrophilic can't move through the cell membrane because it's hydrophobic. But non-polar things that are lipid-based, they move easily through the cell membrane. They can slide right through that phospholipid bilayer. And so this helps it, this lipid membrane around the mRNA helps it get into the cell. And then once it's into the cell, that lipid bilayer will break down and that mRNA will start to be expressed. Well, the gene specifically that they have isolated here and are using in the virus is the one that's for the spike protein. If you've seen a coronavirus image, surely you have after all this time on the TV and stuff, it kind of looks something like this, and it has these spikes. Well, those are proteins that are on the surface of the virus. And so your body starts to produce those proteins. because it's got the mRNA in there, basically. And then as a result, your immune system recognizes those as foreign proteins. And so it starts to cause an immune reaction against them. And then, make memory cells and make antibodies and all of those things, so that if you were to actually encounter a true, real coronavirus, not just the mRNA from the vaccine that you got, then your body is equipped to be able to rapidly fight that. It remembers that, and it doesn't have to make all of the cellular machinery to fight it because it's already there. Okay? So vaccines are important against viruses. All right. Now, we've also are firsthand aware of the terms epidemic and pandemic. I made this PowerPoint several years ago, but we know firsthand what that is now. Epidemic is going to be a general outbreak. It's going to be more localized, versus a pandemic, is global, what we're seeing now. And then the last type of pathogen I want to talk about, we know pathogens are anything that cause disease, okay? So it can be viruses, it can be bacteria. We talk about several, in PBS, we talk about several different types of pathogens. Fungus is a pathogen. Worms, usually adults don't get worms, but like animals get worms. Sometimes kids will get like worms in their belly, tapeworms and stuff. That's a pathogen. But prions are another type of pathogen. And what a prion is an infectious protein. Normally, these are going to be transmitted in food. They typically have a tendency to cause neurological disorders. If you've ever heard of the mad cow disease, I don't know when in my life we had a kind of a bad meat time, and there was some issues with mad cow disease. I remember learning of that, in the news at some point in life. So I don't know if you've heard of that or not, but mad cow disease is a result of an infectious prion. So those are all different types of things that can cause infection. All right. Now, so that's the end of that lesson, but I'm going to jump right on in here into the next lesson. So if you need to pause me and take a break, go for it. But the next lesson is biotechnology. So biotechnology is the manipulation of organisms to be able to make useful components. And so you took the biotechnology survey, hopefully, and worked on that with your family or friends or someone, grandparents maybe even. And you were hopefully you had good discussions with your family about that. But we all kind of have different opinions and different ideas about what's acceptable and what's unacceptable. And so biotechnology is talking about those types of things and the science to do that. So is genetically engineered food okay? How do we genetically engineer food? How do we manipulate things to make these products that we use? How do we make insulin? That's all part of biotechnology. Okay, so biotechnology is kind of the study or the process of it, but actually the action is through what's called genetic engineering. So that is manipulating genes genetically. So, for a long time, we have kind of done this selective breeding. That started with Mendel in the pea plants. Well, probably even before then, but that's an example, a really prevalent example in the 1800s. So, he would take the peas and he would cross them and write down, what traits went together and made what. And he was able to achieve these flowers that looked exactly like he wanted to. And most of us would probably look at that and be like, well, that's no big deal. That's, not very exciting. That's not biotechnology. But in a way, that was like the old school biotechnology. Right? That was selectively breeding things to make them the way that we wanted them to. Even more recent than Mendel, but still kind of, something that's been done for a long time is like breeding dogs. And in Kentucky, breeding like derby horses and race horses. And so they'll take, I don't know if you all have pets that you've paid, some dogs you can pay thousands of dollars for to get a really pure dog of a certain species that's bred to be allergen free and all of these type of things. But that has for many years was done outside of a laboratory. There was no true genetic manipulation, but we were selecting for the traits that we wanted. But now the science has evolved enough that we are able to do that genetically and truly choose these genes. And so that's what genetic engineering is. So in the process of genetic engineering, we often make something that's referred to as recombinant DNA. So recombinant DNA is when you have DNA from two different sources. And it's often combined in vitro, and that just means like in a petri dish, outside of the organism. And then it can later be put into an organism. And so let's take a look and, we'll talk about more about recombinant DNA in just a minute. But you take a DNA from one substance basically and you enter it into another substance, and that is recombinant DNA. All right. So when we look at this process of DNA cloning or gene cloning, we often choose bacteria as one of our ideal hosts to be able to make these genes of interest. So for example, insulin is a really important one. A huge population of the US is diabetic, right? And so many people rely on insulin. And so insulin is made using bacteria as a host. So when we look at bacteria, we haven't talked a ton about the structure of a bacteria. You should be familiar that, bacterial cells do not have a nucleus, and they have one circular chromosome, okay? And so we see that here, that bacterial chromosome. But they also have a second piece of genetic information. So this is a circular piece of DNA called a plasmid. And it replicates separately from the bacterial chromosome, okay? Replicates separately from the chromosome. Bacteria can live without it. You could take it out of the plasmid and it would be fine. So it's not crucial. It is not the bacterial chromosome. It has many fewer genes than that, but it is a secondary piece of DNA there. And so what happens is we would look to like a host, or not a host cell, like a source cell. So let's say this source cell is a human, and we go into the human cell and we get the DNA and we isolate a gene of interest. And specifically, let's talk about the gene that makes insulin, okay? So we take that gene of interest here and we put it into our bacterial plasmid, all right? And then that bacterial plasmid is put into the bacteria. And so every time that bacteria divides, the plasmid is going to copy too. And so we see the plasmid in all of these cells. And then as it starts to express its genetic material, the plasmid is going to express its genetic material too, including the gene of interest. And so we get these proteins that are made here from our gene of interest. And so then we can harvest those proteins and use them to make things like insulin. Human growth hormone is another important one. I had a friend in high school that took growth hormone shots for a long time just because he was kind of a small dude. And so that's an important one. But they are able to do that through recombinant DNA. So this is our recombinant DNA, our plasmid, once it has that gene of interest in it. All right. So this process, sorry, my screen keeps moving all over the place, is called gene cloning because we are essentially just cloning that gene. We're copying that gene many different times so we can harvest this protein. They'll also use those genes to do research on them after they're able to isolate many copies of them. That's a way to make a lot of copies of a gene. You can do it this way through gene cloning. There's bacteria that are made this way to be able to clean up oil spills after an oil spill, like in the Gulf or something. They also will sometimes put genes into plants that gives them pesticide resistance. So there's lots of different things that you can do, okay? So that is gene cloning, amplifying a particular gene or produce a specific protein using that plasmid, the circular DNA molecule that replicates separately from the bacterial chromosome, okay? So let's get into restriction enzymes now. Now you should be a little bit familiar with restriction enzymes. So restriction enzymes, basically what they are is they're cutters, and they cut DNA. Now, they're very specific for the location that they cut. So if we had a DNA molecule, I'm just going to make up one here. Okay. you might have a restriction enzyme that recognizes the sequence GGTT and cuts between it. It would cut between the GG and the TT. So here you would have a cut, and there's no other GGTTs there, okay? So that would be it. You may also have one that recognizes the sequence TCA, TGC, and makes a different type of cut, okay? And let's see if we have one of those, TCA, TCG. Now, I didn't write my second strand here, but you know DNA is going to have a second strand here. Okay, I'm not going to finish that out. So there's two types of restriction enzymes. Ones that make this type of cut are called a blunt cut, okay? And others that make this type of cut produce what are called sticky ends. So you see these sticky ends shown here. This restriction enzyme, whatever it was, recognizes the sequence GAA-TTC. And so it's going to read GAA-TTC. And it's going to cut in that spot. And so these little overhangs are called our sticky ends. And then we can insert our gene of interest into that spot that's also been cut by the same restriction enzyme. So it has the same sticky ends. And then the sticky ends will line up. If you look at those letters, they base pair perfectly. And then we have to seal them back together, okay? And so the sealer, as just was the sealer in DNA replication that sealed those Okazaki fragments or those lagging strands, is DNA ligase. And it seals it all back together to make that one recombinant DNA molecule, okay? So restriction enzymes are different though. The enzyme, maybe this is, they have weird names like EcoR1, BAM-HI, HEND3, whatever. And you don't have to memorize any of them. You would always be told. But maybe this is EcoR1. It recognizes one sequence. And BAM-3 recognizes this sequence or whatever. They all have different sequences that they recognize. And so we use that to our advantage in science based on where we need to cut the DNA. You're going to want to cut it in certain spots based on where your gene of interest is. So you don't want to cut it right in the middle of the gene of interest. So that may eliminate using one restriction enzyme over a different restriction enzyme. So hopefully that makes sense. Restriction enzymes are also important, so they can be used to cut DNA to be able to manipulate it for genetic engineering and making recombinant DNA and stuff like that. But they're also important because they cut up foreign DNA in the cell. And so part of their job is also to recognize foreign viruses and bacteria and things like that in the cell and degrade their DNA as well. Okay. We're going to talk now, shift just a little bit about and talk about biotech techniques. And some of these you are already familiar with. So many of these are going to use the terms that we've talked about here in the past. So the first one is gel electrophoresis. And so gel electrophoresis is used to separate nucleic acids. So we're talking about DNA and RNA here. On the basis of two properties. And so those properties are size and charge, okay? So here's our gel electrophoresis apparatus. You connect it to voltage. You're going to put the negative end at one end, the positive end at the other. Here we have our agarose gel, okay, that's got these wells in the top, and you're going to load the DNA into the wells. So if this is our restriction fragment that we've just done, we have cut it twice in this diagram, and we have 1, 2, 3 different fragments that we've made, okay? And so you would take each of those different fragments, 1, 2, and 3, and put them in a different well in your electrophoresis chamber, okay? And then they're going to run on the basis of size and charge, all right? Well, DNA is a negative molecule, okay? Remember, DNA, or all nucleotides, so DNA and RNA too, are made up of 3 things. They have a 5-carbon sugar, they have a phosphate group, and they have the nitrogen bases. which are the A's and T's and G's and C's, okay, or U's in uracil. So the phosphate group is where it gets its negative charge. If you remember phosphate from biochemistry, it's PO4 minus 3. That's the negative charge of DNA. So we put our DNA in at the negative end because opposites are going to attract, and that'll make it run down to the positive end, okay? So there's the charge that we're talking about. Now, it also separates on the basis of size. So if this was our DNA sample that we were loading in, this one has 4 bases. This restriction fragment is what the pieces are called, has 3. And then this restriction fragment has 7 bases, okay? So if we ran them, if we ran them in an electrophoresis, you would have, I'm just going to draw a new line, a 3, a 4, and then a 7. Oh, I did that totally wrong. Ignore what I just said. I did that totally wrong. You would have a 7, a 4, and a 3, okay? The big guy, the 7, is not going to run very far. He's big, and so he's going to kind of get stuck in the gel. The 4 will be in the middle, and then the smallest guy, the 3, will run the furthest, okay? And so there's where we get that size component, all right? So on Monday, we're going to do a gel electrophoresis lab. So be planning on that to be at school on Monday for the electrophoresis lab. So what you get when you do an electrophoresis is something that's called a DNA fingerprint, or maybe referred to as a genetic profile. So these are used a lot in criminal cases. You can use it for paternity cases. They have lots of applications to them. So if this is our evidence here, and we've got 2 suspects, we would want to convict suspect 2 because he is going to most closely match the evidence, okay? So, but again, before we do this, we have to use our restriction enzymes to cut the DNA to be put into the electrophoresis. So we would take our DNA from the crime scene, treat it with restriction enzymes, and then put it in the electrophoresis chamber, all right? In that process, you wouldn't actually do any of the recombinant stuff, all right? Now, one last technique I want to talk about is PCR. PCR stands for polymerase chain reaction. Remember, polymerase is the enzyme that helps to copy the DNA polymerase. And so in polymerase chain reaction, that's basically what we do. We make many, many, many copies of DNA. You can make billions of copies of DNA in just a couple of hours using PCR. So oftentimes when DNA is collected or obtained, to be able to actually test anything with it or do anything with it, you have to amplify. You get a very small sample, and so you need that sample to be larger, and so they do PCR. Perhaps you've heard of PCR recently with COVID. So if you've had a COVID test that wasn't the rapid test, if you had a test where they had to send it off to a lab like overnight or over a couple of days, that was a PCR test. So basically what they did is they stuck that stick up your nose and got some of your snot with your DNA in it, and they, took that sample and sent it off to the lab, and they ran PCR on it. They copied that DNA hundreds of thousands or millions of times, and then they looked for the specific gene, the coronavirus genes that they are trying to identify. And so, perhaps you've heard of a PCR test recently in that regard. Okay? So, I even skipped that lesson in class. We'll go ahead and talk about this really quick. A DNA microarray assay is one other thing. And so we did a DNA microarray. That's what we did with the pink and the clear liquids that we did with the cancer lab just a week or 2 ago. And so it is used to detect the expression of genes, whether a gene is turned on and expressed, whether it's expressed at too high of a level or too low of a level. But that is another biotech technology that's used in looking at gene expression. All right. So hope you're having a great day, and we'll see you soon.